So this is the fifth lecture in Christoph Koch's and my series of lectures on the visual system and neural computation. And the title of this talk is Optical Imaging of Brains. And I decided, alongside showing all of the wonderful new techniques that have come out over the past several decades, to step back a bit and show why we need new techniques, or why we needed new techniques. And the simple reason is that studying the brain is hard. It's actually very hard. Neuroscience has made a tremendous number of advances over the past century. But we really have hit a wall over the past 30 years, I'd say. And with the techniques that we had, we really weren't making progress at the accelerating pace that one would expect of modern biology. But really over the past decade, the techniques have gotten so much better that neurophysiologists such as myself, who were trying our best to make new breakthroughs in studying the physiology of brains, but we were getting a bit discouraged because things were so hard. But really, I think the most important thing to emphasize for this entire course, and really for the endeavor that we are beginning here at the Allen Institute, is that things are looking up, that the ability to study neural networks in the living brain has just, our ability has increased and the techniques have exploded. So let me go back to the beginning. This is probably the first or second slide that I showed at the very beginning of this lecture series in the lecture entitled The Visual System. And this shows the human visual system from eye to brain, with a great deal of emphasis, of course, on the eye and on the cerebral cortex and the visual cortex, where we have decided at the Allen Institute to make most of our efforts in studying the visual cortex in our 10-year project that's just beginning now. So really, this is not the visual brain, this is the human brain. This is just a picture of the brain. And this, as I emphasized in the first lecture, this is the brain presumably seeing a spot of light. And I spoke for at least half an hour about how a single spot of light might excite the visual brain. But it's just a picture, and it might just as well be a picture of a brain of someone listening to Jimi Hendrix. By the same token that you really can't see the brain seeing, you can't see the brain hearing, and you can't see the brain having very, very intense sensory input and very intense feelings and emotions that might come from listening to loud music that really might move you. It's still the same brain. And this is still the same brain that you might look at of someone daydreaming during a lecture. So what's the problem here? The problem is very simple. It's so simple that it almost doesn't bear saying. Why is it hard to study the brain? Its components are invisible. You can't look at a brain and see its neurons. You can look at a brain and you can see the convolutions on the surface and guess that that part might be important because it's trying to jam a whole lot of things into a small space, hence the convolutions, but you can't see what it's trying to jam into that small space, which is a whole lot of neurons. Not only is it invisible, it's also silent. You can look at a brain and you might be able to look at the neurons, but if you want to figure out what it's about, you can't. The activity of neurons is completely invisible. And with many things in studying particularly the physiology of animals, a big hint about what the physiology of something is how it moves, you know, a heart beating or a muscle contracting. The first approximation, and to a very good first approximation, the brain doesn't move when it's doing what it does. So it's just this hunk of tissue that we're confronted with. We know it's probably, you know, the most important hunk of tissue in our bodies, but how do we study it? And I think the most important fact that I'm going to emphasize now, it's very, very big. You know, it's not so big in absolute terms, but if you think of the individual components, it's extraordinarily big. So let me go through what I mean by that. How big is a brain? A human brain is roughly fits in a box 10 centimeters on the side, and if you do the math and know units, that's approximately 1 liter. The volume of a brain is approximately 1 liter, and I underlined approximately here. I'm going to throw out a whole bunch of numbers, and I'm perfectly happy to be wrong by a factor of 50%, but no more. So these numbers are very round numbers, and it's actually nice how round the numbers are in terms of just remembering all of these things. So a human brain is approximately 1 liter, and if you remember high school chemistry probably, 1 liter is 1,000 milliliters or 1,000 cubic centimeters. So it's a 10 centimeter box, but if you do the math, 10 cubed is 1,000, and one human brain is 1,000 little milliliters. And by point of reference, I think it's 2.5 or 5 milliliters in a teaspoon. So why am I saying this? The important thing is that a human brain contains 100 billion neurons. And it's a number that people throw out all the time, but it's hard to sort of make sense of such a big number. 10 to the 11 neurons, what does that actually mean? So I'm going to do just a little powers of 10 exercise to try to bring that home, how immense the brain is. So let's go down by a power of 10, and it's kind of convenient that the animal that we're studying here at the Allen Institute is a mouse, and a mouse brain is exactly 10 times smaller, or 1/10 the size of a human brain. So rather than 10 centimeters on a side, a mouse brain roughly fits into a cubic 1 centimeter, or 1 centimeter cube, approximately 1 cc. But here comes the 3 dimensions. What does that mean? If it's 10 times smaller, or 1/10 the size, 10 millimeters cubed, or 1,000 millimeters cubed, but let's go back to what I had earlier. 1 centimeter cubed is 1,000th the size of a liter. So the mouse brain is in fact 1,000th the size of the human brain. And the numbers work out pretty well in terms of neuron number, where we had 100 billion neurons in our brains. We have only, but it's still a big number, 100 million neurons in the mouse brain. So a factor of 10 turned into a factor of 1,000, and we have something that's tractable. At least it's tractable for a lot of reasons that don't have to do with its size. We study mice because we have genetic access to mice. We can breed them up quickly. We can put proteins into mouse brains that allow us to study mouse brains. But the other useful fact about studying a mouse brain is it's 10 times smaller, linear, 1,000th times smaller in terms of the number of neurons. 100 million neurons is still an awful lot, and at least today, we're not going to study 100 million neurons at a time. So you have to, today, go down another factor of 10 to something that really, this is another useful thing to put on the surface of our 1 centimeter cube. This is a slice through the mouse brain from the Allen Brain Atlas, the Allen Brain Reference Atlas, which if you're listening to this lecture, go back to how you found the lecture, go back to the Allen Institute for Brain Science website, and take a look at the Allen Brain Atlas. and it's a huge compendium of anatomical facts. Specifically, it has a lot. The reason it exists is to show gene expression in the mouse brain, but along with that, a reference atlas was made where we have, the Allen Institute has 132 of these pictures, and each cross-section through the mouse brain is roughly a square centimeter. And you might be able to see a single neuron there, but you're pretty hard-pressed to see them. Let's go back down by a factor of 10. And this is where things begin to be tractable. This is at the scale where we can actually do systems neuroscience and circuit neuroscience. So how big is a brain? Very big. How big is a cortical module? I'll call it a cortical module, and I'll just define it in the mouse as a cubic millimeter. So a cube, 1 millimeter on the side, a tenth of a centimeter. In terms of volume, that's a microliter. It's the tiniest drop you can imagine, or a very small drop. It's 1 millimeter cubed. And by simply going down a factor of 10 in each dimension, we've gone down another factor of 1,000 in terms of the number of neurons. So here is a circuit I took from the second lecture I gave, the third lecture in this series. This is just a very stick figure diagram of a cortical circuit, and just a bit of it where you see individual cortical neurons drawn by hand in a series of anatomical experiments. But this is clearly not 100,000 neurons. These are just a handful of neurons that were drawn to illustrate what cortical neurons look like. Here we go. This is, again, from the same resource on the Allen Institute website. This is a square millimeter of cortex, and I'm just putting that on the face of this cube. And right here, just on the surface of it, you might see 1,000 or several thousand neurons, but if you fill it out in 3 dimensions, you have 100,000 neurons, and that's a lot of neurons, but it's something that we can begin to tackle with new techniques, particularly optical techniques. Let's go back down another factor of 10 to something that really is beginning to, you can actually see the individual components, and you can really think about them as a decently small number. Here we've gone down, again, a factor of 10 linear from 1 millimeter to a tenth of a millimeter, which is the same as 100 micrometers, 100 millionths of a meter. And that's only 100 neurons that are contained in a nanoliter, that are contained in a tenth of a millimeter cubed. And now we're beginning to get to sizes that you really don't think about in everyday life. A cubic millimeter is a very small grain of rice, or a very large sesame seed. It's something that you could imagine holding in your hand and having just the tiniest bit of heft. 100 microns is much smaller, and it's almost exactly the size, the thickness of a human hair. So if you could imagine taking one little human hair and trimming it so it forms a cylinder that's just as high as it is wide, something that you wouldn't be able to touch with your hands, that's a cubic, that's a nanoliter, a billionth of a liter, and it's a cubic 0.1 millimeter, or a cubic 100 microns. And this is the scale that you really can begin to think of a cortical ensemble, a group of neurons that are next to each other that you could imagine, gee, I want to study this brain. I would like to know what all of those neurons are doing. And that's something that, as you'll see, optical techniques are giving us for the very first time. But let's go back down another factor of 10 before we do that, and we'll get to the absolute unit that. So again, sorry, this is from the Allen Brain Reference Atlas. zooming in pretty much as far as you can go, and you can see here on the face of it, you might see 30 or 40 neurons. If you expanded that out into 3 dimensions, you'd see 100 neurons. Here we go, the final factor of 10. And I'm warning you that I'm not going to stop here, but I'm going to stop here today. But when I talk about nanoscale anatomy and in the lecture called the connectome, I'm going to go down 2 factors of 10 beyond this. But today is not a lecture about the wires between neurons. It's a lecture about individual neurons and how we might study them. This is one neuron. This is a hand-drawn 3-dimensional model of a neuron that was done with electron micrographic techniques that I won't talk about today. But one neuron, plus or minus, in the cortex fits into a 10-micron box. A 10-micron box is a picoliter. So you don't come across pico very often, but it's not a billionth, but a trillionth of a liter. It's infinitesimal. It's 0.01 millimeter cubed. We don't come across anything that's a tenth of a human hair, so it's really hard to think about. And on average, how many neurons are contained in one of these tiny little boxes? Actually, a tenth of a neuron. So you might say, how's that possible? One neuron fits in one of these boxes. Why is there a tenth? And the answer is pretty obvious, that the brain isn't made up of closely packed neurons. It's made up of neurons with a whole bunch of axons and dendrites, which are the connections between neurons, and other things such as glia and blood vessels and blood cells, etc. So that I've been giving the bad news so far. Why is it impossibly hard to study the brain? Primarily because it's invisible, it's silent, and it's impossibly big. And again, it's not impossibly big because it's such a large volume, especially a mouse brain is the size of a pecan or something. But it's impossibly big because it contains hundreds of millions in the case of a mouse, or hundreds of billions of neurons. How can we possibly study that many neurons? And the answer is we can't. But we can begin to think about studying circuits where the number of neurons in a circuit ranges between 100 and 100,000. So that's the bad news. Remember the first lecture, I was more cheerful than I am today about studying the brain. I was giving a 50-year overview about the wonder of studying the visual system. And one of the very first things I did, or the very first thing I did, was say, listen to a single neuron. And for many of you, especially on the web, who haven't studied neuroscience, the sound of a single neuron might have been something new and something very exciting. And in fact, it is very exciting. Here we go again. This is a 50-year-old technique where you can eavesdrop on a single cell in the brain, and you can figure out pretty much exactly what it's doing or what it's trying to do. This neuron is signaling to the rest of the brain a spot of light. And the details don't so much matter, but you really can begin to understand how this neuron works just by listening and watching what the input is like. I'll stop that. I'll stop that and say, electrical recordings are great. They're really what started and created modern systems neuroscience. But they give you not the entire thing. They give you one unit at a time, one neuron at a time. They give you the activity of one neuron. And now things have gotten somewhat better with electrical recording. Rather than listening to one neuron at a time, we might listen to 10 or 100 or even several hundred neurons at a time. How might we do this? So let me go to the next picture, which is from David Hubel's book in the 1980s, illustrating this great technique of microelectrode recording. And now that you have a sense of the scale of neurons, you can interpret this picture here. This is a picture of two things that are shown on top of each other. First, in the background is a bit of brain. It's not the brain of a living animal, but it's the brain of an animal that's been, whose tissue has been processed, so you can see individual neurons. So now you know that each one of these individual neurons is roughly 10 microns, 1/10 of the size of a human hair. And the great technological advance of the 50s and early 60s is the ability to take very, very fine wires that taper down. They start out roughly the thickness of a human hair, and they taper down to a very sharp point that's 100 times finer than that. The point is less than a micron, and the part of that electrical wire that's listening to electrical activity in the brain, that's several microns around. So you can think of the microelectrode, the great technology of the 60s all the way through present time, where the vast majority of neurophysiology is from electrical recordings such as this. And the problem is that this is only one recording. Again, with modern techniques, you can have hundreds of these, but only hundreds of these. And the other very important thing is you can't do what I want to do here. Go from this neuron to that neuron to that neuron to that neuron. There's no way of taking something physical, a piece of wire or a piece of silicon nowadays, and moving it around in the brain, sampling from one neuron after another. You can't look at the circuit and say, I want to know what all of that circuit is doing. So that's the bad news. That's where we were 10 years ago. How can we get out of that problem? And the answer is brain imaging. And really, there's been a revolution in brain imaging over the past 25 years. I've been sort of changing the numbers back and forth. The low resolution forms of brain imaging have been around for 25 years, and the very high resolution forms of brain imaging that I'll finish this lecture on have been around for 10, 15 years. So let's talk about three types of brain imaging. First is the kind of brain imaging that's by far the most known in the popular press and in popular science television shows, etc. It's magnetic resonance imaging, or MRI, or functional MRI. And this is a technique that's used in human beings to really peer inside the brain and not only see the human brain with resolution of 1 millimeter, but in fact see the human brain process information, or see the human brain activated when that brain is processing information. So remember, I said the human brain is very big and it's silent. How do we study all the different parts of the brain? That's with the magic of MRI, which gives three-dimensional images of tissue. It could be your knee, it can be your shoulder, or it can be your brain. Three-dimensional images of tissue. At its very best, it has the resolution of less than a millimeter. So if you remember the little power of 10 exercise that I did at the beginning, if you have resolution of 1 millimeter, that you have 1 million of those spots throughout the human brain. So you're able to look at 1 million different places in the human brain, which is pretty astounding. And not only are you able to look at 1 million different pieces of the human brain, you can actually see the activity of each one of those 1 million parts of the brain. And that's the great excitement of functional MRI. And in fact, Christoph Koch in the next lecture will be telling you about that. It allows you, and the pictures that I think everyone has seen nowadays is parts of the human brain lighting up when the human brain thinks about something or gets angry or sees something. That's all I'll tell you about fMRI, because Christoph will tell you about that. But I just want to say that's all the positive. The negative is just almost by definition something that would have to be true. If you want to study the whole human brain, you're not going to study every single neuron. The technique isn't going to allow you to do that, but even you can't imagine studying 100 billion neurons at a time. It couldn't possibly do that. So each one of those little points in a three-dimensional map of the human brain, each one of those points is a cubic millimeter or 100,000 neurons. So we're not seeing the activity of the computation of each one of these. We're seeing the activity of the average activation of these circuits. So how can we get closer to looking at neural computation of circuits and of individual neurons? And that's with light. Christoph will tell you about the physics involved with this kind of imaging, but it's not with light. But with two types of optical imaging, we can get higher resolution pictures of experimental animals' brains. And the first one, historically, is called optical imaging, usually. That was the title of my lecture, optical imaging. But in fact, for the people in the field, the low-resolution, older version is what we call optical imaging. And this is just taking a regular TV camera or a fancy scientific TV camera and looking at the brain. And I'll tell you about that in the next several slides. What's the resolution of this technique? The resolution of this technique is not 1 millimeter, but 50 micrometers, or 0.05 of a millimeter. So the resolution is much better than the MRI. And the temporal resolution, which I didn't mention for the MRI, is roughly 1 second usually. So in order to hear the crack, 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 crack of an individual neuron, these neural recordings have to be taken at least 1,000 times per second, but typically 10 or 20,000 times per second in order to hear the individual crack of these neurons. The older techniques and the lower resolution techniques for optical imaging do not have this temporal resolution, so they really don't give us information about the very fast neural activity. That's not quite true for optical imaging because there are different versions of optical imaging that include sensing voltage, which can be much faster. But in general, optical imaging as a technique has been measuring slow activity in the brain. And that was a great advance, and I'll tell you about that in the next several slides. Two-photon microscopy is really the reason why I'm here, and I think the reason why reason I'm here, meaning the reason I moved to the Allen Institute to start a large-scale project along with many colleagues to look at neural activity. This new technique of two-photon microscopy has just blown up in the field, allowing us to see things that weren't visible before, and particularly with calcium indicators, and I'll talk about those later. What does this give you? It gives us another factor of 50 or better resolution. So we went from 1 millimeter to 50 micrometers, now to less than 1 micrometer, so less than 1/10 of the size of a neuron. So the resolution is it's too good to be looking at individual neurons. And the resolution in time is several ms, which means milliseconds. So in fact, we can really approach looking at single neurons and individual action potentials or spikes with these new optical techniques, and that's what I'll finish off with. And what's great about these modalities, it allows for repeated physiological and really anatomical studies of the thinking brain. So let's go to the precursors of these optical techniques and really the inspiration for the people who began looking at the brain with optical techniques. And again, like many things, the inspiration for looking at cortical circuits in particular came from the visual system. And again, in this case, the work of Hubel and Wiesel and a colleague in their lab, Simon LeVay. The precursors of brain imaging were anatomical techniques, so looking at not the living brain, but the anatomy of the brain, but with techniques that illustrate physiology. Wouldn't it be great to look at a brain and see physiology? That's what we can do now with optical techniques, but in the 80s, people figured out techniques for doing the next best thing, looking at brain tissue and seeing in that brain tissue something that was related to the activity of neurons. And in passing, in the very first lecture on the visual system, I spoke about ocular dominance as one of the emergent properties, or one of the new properties that comes about in the visual cortex. So what do I mean by that? That the visual system starts with your two eyes, and of course your two eyes are completely independent bits of tissue, and you have two retinas that see the world independently. That information is sent on to the thalamus, and the two eyes are kept separate. However, the information from the two eyes converges onto the visual cortex, and that's obvious for many reasons why we'd want to do that, just to combine all the information rather than half the information, but also it's very important for seeing things in depth, that depending on the depth of an object, that essentially the reason you look, if you look at your finger, and this is, you can do it home or you can do here, if you look at your finger, everything behind your finger is double, or if you look in the far distance, your finger is double. And your brain has to learn to interpret that. How does it interpret things that are double vision? And that's what binocular vision is about. And the very first step in combining input from the two eyes is, in fact, keeping it separate in the visual cortex. And Hubel and Wiesel discovered the idea of binocular separation or combination in the visual cortex by taking electrodes and recording from one neuron and saying, wow, this neuron is seeing only from the left eye of the animal, moving it along, and found that a neuron a few hundred micrometers away was seeing through both eyes, and then moving further along, and now the neuron was seeing only through the left eye. And they discovered that the dominance of one eye or the other went back and forth as you marched across the cortex. And they mused, how must this be organized? Maybe there are stripes of one eye, the other eye, one eye, the other eye on the surface of the cortex. And it took more than 20 years for these stripes to be demonstrated anatomically. I won't tell you the details, but this is a wonderful picture that was taken in the 80s, where the light regions are where input from the left eye is, the dark regions show where input from the right eye is. And you get these beautiful stripes, largely stripes of right eye, left eye, right eye, left eye. That was a lot of work. It was many years trying to demonstrate this one fact. And the field just was hoping for a technique that would allow people to see that in the living brain. And a few brave people decided, let's just point TV cameras at the brain. And those brave people were two main groups, Gary Blaisdell at University of Pittsburgh, and Miriam Grinwald at the Rockefeller University. And pretty much simultaneously in the 80s, they did exactly that. They took TV cameras, took pictures, took movies of the surface of a brain, and had the very, very simple idea. Let's take a movie of the brain of an animal looking through its right eye. Let's take another movie of the animal looking through its left eye. And let's compare them. And the simplest way to compare two movies, or really two pictures, is to subtract one from the other. And as soon as they did that, they got, in one brief experiment, a picture that looked exactly like the painstakingly difficult anatomical experiment. They got, so this is an animal's brain, this is a TV camera, this is a bunch of computers, and back then, computers were simple computers in the 80s. And just by subtracting images, they got these beautiful stripes. And you say, that's wonderful, but we sort of knew that from the anatomy. Well, the answer is yes, it is wonderful, but it's something special, it's something different. Because you're doing this in a living brain, it's not one experiment that you do. You can do any number of experiments. You can take movies of the brain when the animal is in all sorts of different states. The simplest set of movies you can, so this is one picture of the brain. It looks like a brain. It's nothing special. This is a difference image of pictures of the brain looking through the right eye, and from that was pictures of the brain were subtracted when the animal was looking through the left eye, and you get these beautiful stripes. So you might get those data in 10 minutes, but then you can do another experiment. You can do something I spoke about even more in the first lecture. You can look at the property of orientation. You can have the animal looking at vertical bars for 30 seconds, and then have the animal look at horizontal bars for 30 seconds and different obliques. And you can do a fancier form of subtraction of all these images, and you can essentially see which portions of the brain essentially light up, or in fact darken, but become a different color, bright or dark, when the animal sees different stimuli. And in order to show this complicated idea, they had to make color codes where the red regions of the brain are those that are most excited by horizontal bars. The green regions are the regions of the brain that are most excited by vertical bars, etc. And this actually answered a very pressing question in the field. We know we have a map for ocular dominance that looks like stripes, but there's another map going on in exactly the same part of brain. What's the relationship? How are these different types of vision mapped onto the same part of the brain? And the answer is nature, at least in some species, found the solution of having the different eyes arranged in stripes and having the different orientations really arranged in different regions that are tiled in a certain way. So one is extremely orderly and one is much less orderly, and they're superimposed. And that actually was quite important for people's thinking about cortical computation. But still, this was exciting. This was a great advance in terms of understanding cortical computation and how different computations were arrayed across the surface of the brain. But what was the resolution? Remember, the resolution was roughly 50 microns. Let's make it 100 microns because we like powers of 10 in this lecture. 100 microns, you might remember, a cube of 100 microns is 100 neurons. And each one of the independent bits of information from these pictures was at least 100 neurons, probably several hundred neurons. So this wasn't good enough for looking at individual neurons and seeing what they do in the living brain. That changed radically in the past 10, 15, 20 years, and it changed for two reasons. And the first reason is the explosion of the use of fluorescent molecules in studying the brain. What is fluorescence? Fluorescence is simply the means by which some types of molecules can absorb light of one color and transform that light in ways that I won't talk about and emit photons of a slightly different color. We're all familiar with fluorescence really from black lights and fluorescent paints, that you can take ultraviolet light that we are essentially blind to, and fluorescent paint will absorb that light that we can't see and then emit another kind of light, usually green, red, or even blue, and you can see these bright colors coming from nowhere. Well, they're not coming from nowhere, they're coming from light of a different color. Fluorescence has been used for quite some time in looking at neurons, but there was just a radical revolution in looking at individual neurons with fluorescent proteins. And the granddaddy of fluorescent proteins was green fluorescent protein, which is a molecule from jellyfish, a protein from jellyfish that's encoded by some DNA. And by putting that DNA into other species, you can make cells in other species fluoresce. And when you put the molecule in neurons in particular, it makes neurons fluoresce. And this is a lovely paper from Jeff Lichtman and Josh Saines's group then at Washington University, where they put green fluorescent, yellow fluorescent, and even red fluorescent proteins in individual neurons or individual types of neurons, and could take these amazing pictures of groups of neurons, single neurons, and even very high resolution pictures of dendrites, of a little bit of a neuron. This is a dendritic shaft with a bunch of little protuberances where synapses are formed called spines. And it really allowed people to look into the brain and see things that hadn't been seen in quite such beautiful resolution with the old stains. But more importantly, it allowed individual neurons to be targeted, to be visualized. That's one revolution, fluorescence microscopy. But really, the functional imaging took off with fluorescence microscopy with a brand new form of fluorescence microscopy that came into use roughly 15 years ago. And this is the hardest slide in the lecture. It's a bit of physics, and but I hope it's understandable, and I think it's quite important. So how does fluorescence microscopy work? I've already told you the basic principle. A light of one color comes in, and light of another color is emitted. And there are two forms of fluorescence microscopy in use: single photon, or a conventional fluorescence microscopy, or two-photon or multi-photon fluorescence microscopy. Let me start with the much easier one, the conventional one, where light, blue light, or even ultraviolet light that we can't see, courses into the world, and in this case into a piece of brain, and blue photons excite fluorescent molecules, and those fluorescent molecules emit photons of a different color. And by far the most common version of that is, again, a blue or ultraviolet light coming in and green coming out with a green fluorescent protein. And those lovely pictures that you saw are largely green or yellow fluorescent proteins in individual neurons. So why would you, and that's a very simple technique that microscopes in every biology lab have fluorescence capability, so you can see these beautiful green, yellow, or red fluorescent molecules. Why do you need a different type of fluorescent microscopy? And the answer is, this conventional type of fluorescent microscopy really just isn't very good for studying living neural tissue for several reasons. Let me first give the outline of two-photon microscopy. Instead of exciting one of these molecules with blue light, exactly the same molecule can be excited with red light, a very special kind of red light that I'll describe in a few minutes. But let me just say, for now, these molecules can be excited with red or even infrared, invisible light. Why is this important? It's important for two reasons. Two-photon microscopy allows imaging in vivo in the living brain with single-cell resolution. And why is that true? First, red light or infrared light goes into tissue, and there's much less scattering. What do I mean by that? The simplest example is just by referring to the well-known fact that the sky is blue. Why is the sky blue? The sky is blue because blue photons are easily scattered, even by small particles in the atmosphere, which seem essentially invisible. But if you go through enough air, essentially, blue photons get scattered all over the place. And these blue photons that are not coming at you straight from the sun, but in fact are being scattered all over the atmosphere, that's what makes the sky blue. And that's lovely if you want to look at the sky, but it's terrible if you want to do microscopy. If you're trying to use blue light in a milky tissue, it just goes everywhere, and it's a mess, and you can't see very deep. Infrared light goes through tissue much more easily. It just penetrates many times, or many times further without scattering. So one can send this red light into the brain up to 0.5 millimeter or even more. So that's one simple reason why it's good to do microscopy with red light. Here's the complicated, but really the deeper and more important reason. And it goes under the name of nonlinear absorption. And what do I mean by that? It's not the amount of absorption of these molecules isn't proportional to the amount of light going in. It's proportional to the amount of light going in squared. That might seem like a small fact, but it's the big fact. Just regular amounts of red light just won't have a chance of exciting these molecules. But if this red light can be extremely bright in a very small place, and by extremely bright, I mean extremely bright, really at the level of the surface of the sun bright, then these molecules can be excited by the red light, and these green photons are emitted. You can imagine that it's not a very good idea to excite the brain with as much brightness as the surface of the sun. And the only way you can get away with this is two things. The pulses of light are infinitesimally brief, less than a trillionth of a second that the light is on. And not just that, it moves very quickly. The light has to move very quickly. And the way to get both of those things working is to have a laser that sends out this red light, but it sends out this red light in very short pulses, 80 or 100 million times per second. So we have these individual packets of light, bundles of light, that get turned on for a very short amount of time, and these packets of light are sent into the brain 80 million times per second. What does that do? And here's the hardest part of this hard figure. It's from a paper by Zipfel et al. From Webb's lab in Cornell, where this microscopy was invented. This is sending light into a little well of fluorescent liquid. And if you send blue light into a well of liquid, it excites the fluorescent liquid everywhere, from the surface of the liquid all the way down. It's being focused down to a point with a microscope objective. But you can see that we have these green photons coming at you from every direction, and not only from the point that you're interested in, where it's focused, but all along the surface. Look at this picture. It almost looks like a smudge on the photograph. This is one spot where the red light is bright enough that it actually excites the fluorescent molecules and green comes out. So it's like a magic wand that you can point for a second and say, I want to look right here. And I want to do it just for the tiniest fraction of the second and then move on. Let me zoom that in. So this point is less than a cubic micron. It's tiny compared to a neuron. It's even small compared to most components of neurons. And let me zoom into that a bit to really give the flavor of it. One photon microscopy, you have photons everywhere. And you can focus the light, but you have a lot of what's called out-of-focus fluorescence. Two-photon microscopy, you have a red light, much like my laser pointer, right there, flashed on for a trillionth of a second, and it makes a spark. It really makes just an infinitesimal spark in the brain. And what does that have to do with imaging? And the answer is, it really isn't imaging yet, but it's really, remember I set up early on the thought of taking a microelectrode, which is sitting in one cubic micron and listening away. Wouldn't it be great to move that electrode all over the place? This is the magic wand that does that. This is a spot of light that interrogates the brain by seeing how much fluorescence comes out, and you can move that all over the brain in order to make images, 80 million times a second. So who do we have to thank for two-photon microscopes? We really have three people. Maria Geppert-Meyer, who is a physicist who won her Nobel Prize in physics, not for this, but she won her Nobel Prize in physics for the shell structure of atomic nuclei. But I guess in her spare time, she thought of the idea of two-photon excitation, that theoretically it was possible to excite fluorophores, to excite molecules, not with one bit of light, one photon, but two photons. So, Nobel Prize, and as something totally separate, she did the foundation of two-photon microscopes. Roger Chen, who really created the field of calcium imaging by engineering both chemicals and then engineering, in fact, proteins that don't only fluoresce, but they get brighter or darker, usually brighter, whenever a neuron is active with calcium. And Roger Chen also won the Nobel Prize in chemistry, not for this, but he won the Nobel Prize in chemistry just for the green fluorescent protein, along with two others. But he really created the modern field of calcium imaging. And the third thing, so you have the excitation, you have the calcium indicators, and then you have the idea of focusing one point of light and moving it around. And that was the idea of Winfried Denk in Watt Webb's lab in Cornell. When Winfried did his postdoctoral fellowship in Cornell, he, in Watt Webb's lab, invented the two-photon microscope, which is essentially taking a laser beam and scanning it very quickly in order to make microscopic images of fluorescent molecules. And he applied that to the brain in David Tank's group at Bell Labs subsequently. So we're almost done. And really, I've just been telling you why we needed optical imaging, how it worked. And now finally, just in the last few slides, I'll show you the new techniques at work. And in fact, you've seen much of the last few slides, but I think it's in a much richer context now. So I showed you how conventional optical imaging allows you to see functional architecture. It allows you to see the input of the two eyes. It allows you to see regions of the cortex that are excited by different orientations. But again, I've said many times, it wasn't able to do that at the level of single neurons. With two-photon microscopy and these fluorescent molecules that get brighter when neurons are active, with those two those two bits of technology, in the past decade or so, we've been able to, the field has been able to look into cortical circuits. And now this is, I think I called it a cortical ensemble of roughly several hundred microns, several hundred neurons, and look at these individual points of light, and those individual points of light are neurons, and some of these neurons respond to direction in one, some of these neurons respond to stimuli moving in one direction, like this, and other neurons respond to stimuli moving in the opposite direction. And again, that's showing something that we already knew, plus or minus, functional architecture, but at the scale of single neurons. But of course, now that we're able to essentially move our electrode through the brain and sample the physiology of neurons, we're able to look at things that really no one has ever seen before. And these are just two examples of what I showed you was almost a decade old now, a movie that's almost 10 years old. Here are just two examples of experiments that are going on every day here at the Allen Institute and at laboratories all over the world, where this is looking at an ensemble of several hundred neurons inside a living brain of a mouse. And the neurons in this mouse brain have a genetically encoded protein that gets brighter. The fluorescence increases every time the neuron fires, and here you can just see at much higher rates than the previous movie, lots of neurons just firing away. You can really see neural activity at the level that matters, individual neurons. And even it's possible nowadays not just to look at individual neurons, but to see the activity of parts of neurons, and the parts of neurons that really matter, such as the axons that are sending signals from one neuron to the next. So really, it's a technique that it's beyond one's dreams. The dream 15 years ago was to take an electrode and look at any neuron or all neurons at a time and watch the activity. Now it's even gone beyond that to something that I wouldn't have imagined, the ability to look at individual synapses and see the activity of either the presynaptic or the postsynaptic element of those synapses. So I'm just going to finish on a slide that I've shown you before to really say that, or to argue that we're at a place that is unique, certainly, but very, very exciting in the history of neuroscience, that we're able to not just look at 1,000th or 1/10,000th or 100 billionth of a thinking brain. We can look at entire ensemble of neurons in a living brain and try to, with experimental and computational tools, try to understand how neural computation comes about. And again, I like to use this slide just to force, but to really illustrate the idea that if you want to understand a computer, you can't just look at a computer and expect to see computation going on. It's important to use all the tricks in the book, and one of the tricks in the old days was looking at a light panel that instantaneously showed the state of the computation in a computer. And it's hard to say what the meaning of each one of these flashes is in the brain, but it's hard to say that you could hope to understand neural computation unless you could at least see these flashes. Another way of saying it is, I think to understand cortical computation, it really seems to be necessary to be able to look at all of the components. Whether that's sufficient or not, that's the challenge of the coming decades. So in order to think of understanding neural computation, it's important to interrogate all the players in that computation, each one of the neurons, and to test models of how these neurons may communicate to each other and how they might encode information. So I think I'll stop there. Thanks.